Now that we've discussed how old could have increased in complexity from the first protocells, let's discuss the ecological implications of this. There is an ongoing debate as to whether the first organisms obtained carbon from abiotically produced organic carbon in the environment, i.e., they were heterotrophic, or produced themselves from carbon dioxide, or were autotrophic. Given that the first cells came into being in the first place, there had to be biomonomers like lipids and nucleic acids being produced abiotically in the surrounding environment that the protocells would absorb. However, since we know that core metabolic pathways such as glycolysis, the TCA cycle, and even the carbon-fixing Wood-Youngdahl pathway have abiotic analogs, protocells would also be able to take up small organic molecules to be then synthesized into biomolecules, or even produce these organic molecules from carbon dioxide. To see how having a membrane in a variable pH environment would facilitate this, see the Origin of Life video series. Although, this organic carbon would be produced both endogenously and exogenously. As the membranes became more rigid, it would be harder for biomolecules to cross it, and the cells would become increasingly dependent on intracellular carbon fixation and synthesis reactions. This would become increasingly advantageous to the cells as biocatalysts evolved and gave the molecules the correct chirality. Because most parts of any serpentinizing system where abiotic carbon fixation takes place are alkaline, most of the inorganic carbon precipitates out as calcium carbonate, limiting the rate at which organic carbon can be produced. Furthermore, as biocatalysts such as enzymes became more complex, it would facilitate a more constant synthesis of organic carbon from carbon dioxide and hydrogen, meaning that as old evolved over time, it probably went from a mix of autotrophy and heterotrophy towards being autotrophic. While the Wood-Youngdahl pathway bears strong resemblance to abiotic Fischer-Tropsch type synthesis reactions, it is carried out by enzymes in modern organisms. It starts with a hydrogenase enzyme taking electrons from a hydrogen molecule and shuttling the electrons to a formate dehydrogenase, which reduces carbon dioxide to formate. These enzymes could have descended from an early transmembrane peptide with an iron-nickel-sulfur cluster bound to cysteine residues, along with several iron-sulfur clusters. Here, the iron-nickel-sulfur cluster would oxidize a hydrogen molecule, and the iron-sulfur clusters, or ferredoxins, would facilitate the flow of electrons to a bicarbonate, which would then be reduced to a formate. The formate group could then be phosphorylated, forming formyl P, and be bound to another organic molecule, or be reduced further, phosphorylated, and then attached. Folate, or vitamin B9, is often used in the transfer of single carbon groups, and in the Wood-Youngdahl pathway, a modified version of it, known as tetrahydrofolate, or THF, is used where it takes a formate phosphorylated via an ATP. Through a series of redox reactions, the formyl group is reduced to a methyl group by methylene tetrahydrofolate reductase. In early organisms, the formyl group could be reduced by taking electrons directly from the progenitor of the hydrogenase enzyme as it oxidized more hydrogen, or from free-floating electron carriers, which took the electrons from the prehydrogenase. This carbon could then be transferred to nucleotides or amino acids. So far, I've described the west or methyl branch of the Wood-Youngdahl pathway, but there's a second east or carbonyl branch, which involves reducing a carbon dioxide molecule to carbon monoxide. The carbon monoxide, methyl THF, and coenzyme A, or vitamin B5, are then reacted to form acetyl-CoA. How did this branch come about? Well, some carbon monoxide dehydrogenase enzymes possess a copper cofactor, and copper sulfides are the third best mineral catalyst at reducing carbon dioxide to carbon monoxide. The best were cadmium and silver sulfides, but these are far less common than copper. So, a copper-sulfur cluster bound to cysteine could have initially produced carbon monoxide. The acetyl group is bound to the coenzyme A by a high-energy thioester bond, which is hard to produce. The advent of carbon monoxide, with its reactive nature, could have allowed this compound to form. The acetyl-CoA synthase enzyme will initially use a cobalt ion to facilitate the transfer of the methyl group from the methyl THF to the carbon monoxide, which is then reacted with coenzyme A to form acetyl-CoA. Acetyl-CoA synthase itself could have evolved from amino acids, which acted as chelating agents for the catalytic cobalt ion. Three other pathways with abiotic analogs are the TCA cycle, glycolysis, and the pentose phosphate pathway. This suggests that they were initially carried out by abiotic mechanisms in the first cells, and as time progressed, biocatalysts, ribozymes, and enzymes evolved to facilitate the reactions. This would increase the rate of the reactions, the production of minor products would be rendered negligible, and it would ensure that the products had the right chirality. Whether the pathways went forward or backward would depend on the abundance of reactants and products, and enzymes wouldn't change this because they don't alter the equilibrium conditions. They only increase the rate of the reactions. With carbon being fed into these pathways in the form of acetyl-CoA, the reactive nature of the high-energy thioester bond the acetyl group is bound to would push the reactions forward in a unidirectional manner. Furthermore, FADH2 and NADH, derivatives of vitamin B2 and B3, would facilitate this by acting as electron carriers and allowing for greater redox coupling. Methanogens and acetogens use the horseshoe version of the TCA cycle, which is used to produce the precursors of amino acids. And early life could use the reductive version of the pentose phosphate pathway to produce ribose. If the first ribozymes were ligases, phosphorylating organic molecules to be fed into the pentose phosphate pathway wouldn't be a huge leap because sequence ligation is already a variety of phosphotransfer reaction. So, if the RNP world existed, the ribozymes, which catalyzed the early pentose phosphate pathway, would have had a smaller peptide component than most other ribozymes. Many enzymes currently involved in glycolysis and the pentose phosphate pathway have magnesium and zinc cofactors, suggesting that they evolved from ribozymes. Conversely, many of the enzymes which partake in the TCA cycle have iron-sulfur clusters, suggesting that these were built around inorganic mineral catalysts. So far, we've talked a lot about carbon metabolism, but another major biophilic element is nitrogen. The alpha keto acids produced via the TCA cycle are converted to amino acids by aminotransferase enzymes. These possess a PLP, or vitamin B6 cofactor. Can this reaction be traced to the origin of life? Well, it's hard to say because a mechanism by which Strecker reactions could be catalyzed by copper and PLP has been described, but we're not sure whether it uses the same mechanism as aminotransferase enzymes. It appears that two groups of aminotransferase enzymes evolved independently around PLP, one with a type 1 fold and the other with a type 4 fold. Aminotransferases are organized into four classes, and the type 4 fold only contains class 3, which produces branched chain amino acids. The remaining classes are of the type 1 fold variety, and class 4 produces alanine and serine. Class 1, the most diverse class, produces all other amino acids, while class 2 aminates non-amino acid organic molecules. We know that all four classes had arisen in old and had diversified by the time of Luca, but not to their current extent. How the nucleobase synthesis pathway evolved is not well studied, but the difficulty in producing the glycosidic bond, which binds it to the ribose abiotically, suggests that the biotic synthesis pathway evolved de novo. Earlier in this series, we discussed what the primordial energy of metabolism of early life would be like, but how did energy metabolism as we understand it come into being? The ATP synthase enzyme is embedded within a plasma membrane. There is usually a greater concentration of protons outside the membrane than inside, and there is a pore in the ATP synthase enzyme. When the protons cross the gradient from a high to low concentration, they generate a proton motive force, which the ATP synthase uses to power the production of ATP from ADP in orthophosphate. One compelling hypothesis is that the ATP synthase enzyme started out as an ATP-powered pump. When life began colonizing the oceans, it would have to contend with higher concentrations of sodium ions and protons. The ancient oceans had a pH of around 6. And this pre-ATP synthase would push these ions out against the concentration gradient. This would help maintain cellular homeostasis, but increase the demand for reactive phosphorus species. One way to alleviate this would be to use the carbon fixation pathway, and acetogens possess a rudimentary electron transport chain tied to hydrogen oxidation and carbon reduction. Here, ferredoxins shuttle electrons from hydrogenase enzymes to an enzyme complex. Once there, NAD or FAD are used as electron acceptors, and the energy from the difference in redox potential is used to pump sodium ions and protons out of the cell. The resulting NADH or FADH2 shuttle electrons to formal THF to reduce it to methyl THF. Early organisms could use a similar mechanism to pump ions out and relieve the high energy phosphate demand of the cell. The carbon fixation pathway requires ATP, but since the cell has to use it to grow anyway, it may as well use some of the energy from the reaction. However, instead of directing acetyl-CoA into biosynthesis pathways, it can be reacted with orthophosphate to form acetylphosphate, which can then be reacted with ADP to form ATP and acetate. This acetate would be inert and couldn't be fed into any pathway, but now that the ATP is regenerated, the cell could pump ions without requiring high-energy phosphates. Eventually, there could be a strong enough cross-membrane gradient that the reaction of the pre-ATP synthase enzyme would be reversed. Ions flow in and power the production of ATP, giving rise to the ATP synthase enzyme. This would be a major breakthrough for life, as it would no longer be tied to sources of high-energy phosphates, and metabolism can instead be powered by redox chemistry.